This video demonstrates the principles for taking a capillary blood lactate sample. This is a skill that's essential when performing a lactate threshold test. Before we take a capillary blood lactate sample, there are some health and safety considerations that are needed. An important factor is the risk of blood-borne viruses such as HIV and hepatitis. Therefore, we need to take some steps in order to ensure that both the researcher and participant are safe. Firstly, hands should be washed thoroughly before and after sampling. This can be done using alcohol gel or a similar product. Cuts and abrasions should be covered using a suitable dressing before sampling. Gloves alone do not provide sufficient protection. Gloves are required at all times. These need to be changed between participants and also if potential contamination has occurred. Moreover, a new needle device must be used for each puncture that's made to prevent infection. This video will show the procedure of taking the capillary blood sample at rest. The first step, the researcher cleans the participant's finger with an alcohol swab. The researcher then takes a punch device, in this case a self-retracting lancet, and places it on the participant's finger. This is spring-loaded so it punches the skin and then immediately retracts to prevent any risk of skin prick injuries. The researcher then immediately disposes of this. The first part of the sample is removed using a tissue. Then using a collection tube, 20 microliters of blood is collected. Lastly, the participant is given a tissue to stem the flow of blood. Any used tissues are then disposed of in the relevant clinical waste bin. The capillary tube is then placed in an Eppendorf that contains a reaction solution. It is then thoroughly mixed. The sample is then placed into an automated lactate analyzer. In this case, we're using the EKF Biosyn C-line. This device then measures the lactate concentration within the sample. Remember, resting lactate concentrations should be around about 0.8 to 1.5 millimoles. In this example, our resting measure was 1.41 millimoles. The sample can then be disposed of and any surfaces that may be contaminated thoroughly cleaned.